Welcome to this talk that I've been looking forward to for a long time, Dr. Sabine Hazan, all the way from California, where it's 10 o'clock in the evening there and it's 6 o'clock in the morning here. But Sabine, thank you for extending your day for us.
Of course, my pleasure. I'm a night owl anyways, so it's good.
Okay, good. So basically, tell us a little bit about yourself. You're a gastroenterologist and you're researching microbiome. Before microbiome came along, what was your kind of day job?
My day job was doing clinical trials for pharmaceutical companies and bringing products to market. That was my day job. So I was the girl that was basically recruiting patients for different protocols from C. diff to whatever. So it was interesting.
And were you doing clinical work then or as well?
I'm sorry?
Were you doing clinical work as well?
Yes, I was doing, so I started off my career as a gastroenterologist, obviously. I finished my fellowship and my attending said, I got the Dean's Research Award at University of Florida, and my attending said, you're really good at research, you should really keep it up. And I wanted to be in the trenches, doing colonoscopies, taking care of GI bleeders, so I was like, nah, I'm just gonna do clinical GI. And then, 2 kids later, clinical GI was with a husband that's a cardiologist, became a little bit tough, the call and taking care of the kids and juggling. So I went back, I kind of was doing 90% GI, 10% research. And then over the years with 2 kids, it became 90% research and 10% GI. And so that's been my path, essentially.
Brilliant. And when did you first hear the term microbiome?
Well, microbiome, probably at the beginning, right? Because, for all of us, it was fecal material, right? At the beginning, because we were doing fecal transplant, the process of taking stools from a healthy donor and putting it in a patient with C. diff. And so, probably a couple decades ago, without saying my age, but yeah, a long time ago. I remember, in fact, actually, I remember being a fellow at University of Florida and going through the posters, and my friend, Dr. Neil Stolman, said to me, the future is in sh.t. Look at the paper, look at the posters of Dr. Tom Barodi. They're using fecal material to fix C. diff. I mean, this is the future. And I said, please don't make me play with poop. I don't want to go there. And now look at me. I'm like deep into it. So, careful what you say. Indeed.
I've come across C. diff a few times. It's got a rather characteristic smell, but if you don't mind giving us a bit of background, what is C. diff?
So C. diff is a disease basically where people have severe diarrhea.
Yes, Clostridium, isn't it? It's a type of
Clostridium difficile, and by term, which is French for difficult, it's a difficult bacteria to get rid of.
Yeah.
Patients typically were in the hospital. However, in the last decade and a half, we started seeing a lot of young people. And one wonders, were they consuming food like meat from a cow that had a lot of antibiotics, and possibly that's how they got, C. diff. But essentially, C. diff is this microbe that's very difficult to fix. I was the girl that was doing clinical trials to try to kill it. So when I couldn't kill it with the conventional antibiotics like vancomycin, Flagyl, and then, came along fidaxomicin. I did all those clinical trials, fidaxomicin, monoclonal antibodies, spores. And so when you couldn't fix it with the conventional therapy, you would go into clinical trials. And then when I couldn't fix it with clinical trials, then I basically would do fecal transplant, which is basically taking stools from a spouse or a kid or a kid of a person, and then basically putting it in a person's colon. And, the field has expanded. So the beginning of the microbiome really was this discovery that you could take stools and put it in the colon and change a disease process like Clostridium difficile. I think Dr. Barodi, Tom, really took it to the next level of, could it be useful for Alzheimer's? Could it be useful for Parkinson's? Could it be useful for autism? My case, the N of 1, where I had a patient with C. diff that I was doing fecal transplant and had Alzheimer's, and he improved his Alzheimer's. So, it kind of gets you into the path of seeing, well, is there something in the gut? What are we doing? 2 cases of alopecia areata from Dr. Colleen Kelly grew hair, right, after fecal transplant for C. diff. So when you see these cases, but of course, nobody can reproduce that. And then you kind of ask yourself, well, is it donor dependent? What's going on there? So that was my path, essentially, really going after a microbe C. diff, trying to kill it. When I couldn't kill it and I couldn't succeed in saving my patient, I would do fecal transplant. And then fecal transplant, really discovering there's so much more to stools than stools, really. So it was fascinating.
So you're actually using the microbiome, basically the bacteria from someone else.
Yes.
Recolonizing the infected colon, putting the good guys in, and they kind of edge the nasty bacteria out. Is that kind of what's happening? Is it?
I don't think you could say it's good guys. I think it's good guys and bad guys. I think it's a consortium of poop, which are microbes that you put in. I think, the future is going to be more precise. We're going to be a little bit more precise in what we put in. But right now, we're at the level where we're basically taking stools from a healthy donor and putting it into the colon of a person with C. diff. What was fascinating with C. diff is that's a relatively easy condition to fix with fecal transplant. You could do it via NG tube. You could give capsules, or Dr. Brody calls it crapsules. Yep. You could do via rectal. You could do via colonoscopy. And the success is pretty high, above 90%. It's when you try to do it for other conditions and try to use it, it's a little bit more complicating.
But the procedure itself, so you basically just take some fecal material that's been, obviously you screen people for infectious diseases, presumably you check for HIV and hepatitis and...
Yeah, you have to check for a lot more. So it used to be, we would just check for C. diff and E. coli and Giardia and, the common parasites and microbes. But then there were a couple cases of people that died from fecal transplant. And so the FDA demanded that we look for vancomycin-resistant E. coli, carbapenem-resistant E. coli, etc. So it's a little bit more detail. The testing is very long. It's about 11 tubes of blood, and it's about 4 samples of stools just to make sure that it's clean. And so you can't just take anybody and say, oh, you know what, they look healthy, and therefore I'm just going to use their poop and that's it. No, it's pretty, I'll give you an example. I'm supposed to do 2 cases tomorrow morning of people with C. diff. I was supposed to do them, and they were on vancomycin. I was about to do the case, and the 2 donors ended up, one had E. coli, the other one had Giardia. So we had to cancel the cases. So, and these were people that basically did not want to use a pharmaceutical product that's available. So, they wanted their family members. So it becomes expensive when you do that, because you have to test every single thing. And you have to test within that week period that you're going to do it to make sure that you're kind of getting the clean stools. But, here we are, Monday, and the 2 cases are actually canceled because, unfortunately, it's not that easy.
But presumably if you're in, say, a spouse situation or a parent-child situation, you're sharing your bugs all the time, aren't you?
Yes. But you have to remember the person that has C. diff is immunosuppressed, right? So are you giving a microbe where one spouse is strong and resilient to another one, and therefore that's immunosuppressed? And are you going to potentially, harm that person? So I think it's important to test. It's important to be very careful. To me, in my lab, we go above and beyond. We have a genetic sequencing lab where we're doing research, and we're even testing for COVID, because
in the stool.
In the stools. That was my main focus during the pandemic, was looking for COVID in the stools, because I said, I'm transplanting patients. What if I'm giving COVID at the same time, right? So to me, it was important to develop an assay. Unfortunately, it's under research assay. It's not an approved test, but at least it gives me confidence that my patient doesn't have COVID, my donor doesn't have COVID. And we test, obviously, nasal that the FDA requires, but typically.
Yeah, the normal respiratory test.
Yeah, but I think there's a need for looking at the stools, because personally, you could be, I've seen so many cases of nasal negative and positive in the stools, and those are your people that have long COVID, that have digestive problems, that are still persisting in having COVID in their gut, creating havoc. So I think, I'm going to have to fight with the FDA to get that test approved, because I really think I don't know how people can treat long COVID without knowing whether you still have COVID in the stools or not, in my opinion.
Just before I ask you, I want another question about COVID, but the fecal transplant can be as simple as getting some poo.
Yes. So it's basically, you want to create, and you want to give it, you want to process the stools to keep it as anaerobic as possible.
All right.
So because I prefer fresh stools, because that, has a better rate of engraftment, in my opinion. And I think, most GI doctors that do fecal transplant or intestinal microbiota, like Dr. Alex Karutz likes to say, you still want to have as fresh as possible.
And then it's just like giving an enema. You just basically put a tube up.
Yeah, I do it via colonoscopy. We basically wash the colon. We make sure, we give if for C. diff, we give them vancomycin before to kill off everything. It's really, think of it as gardening, right? You've got a whole garden full of weeds. You're removing the weeds, and then you're turning the earth, and then you're implanting the seeds, and you put water and sun, and then your fruits are going to grow. So it's the same kind of thing with fecal transplant, where you're basically killing off all the bad stuff, and then you start fresh by cleaning the colon completely, and then implanting a new microbiome, a new fertilizer, let's say, to start implanting into the colon. That's the best way to kind of, describe it.
So you kind of clean the colon out. The patient presumably doesn't eat for 24 hours. You give them a laxative to clear it out.
Yes, it's exactly like a colonoscopy, and then we go in there and we wash the colon and make sure, and so you want to make sure that everything is clean, and that when you're implanting, you're just putting in the microbes so that they could just start growing. Yeah. So obviously,
so you physically wash the colon out during colonoscopy.
Yes.
Is that well tolerated by the patient?
Yeah, it's this is something we do anyways when we do colonoscopies, not to miss polyps. So this is not new in GI. I think it's important to have a good look at the colon, at the colonic mucosa, and it's important to clean the colon before you implant, in my opinion. Yeah,
absolutely. So next question about long COVID then. In your experience, people with long COVID are still secreting active whole virus.
Some people we've discovered have whole virus in their stools, like up to a year after having had COVID. So if you don't take care of that, you're not going to fix the problem. So, long COVID to me is defined as a couple of ways, right? It's either the virus is still persisting, you've had the virus and you took some medications that killed the virus, but also wiped out your whole microbiome, so you never really come back from that. Or you started off with dysbiosis to begin with, got COVID because of the dysbiosis, and then essentially are having a hard time going back to pre-dysbiosis, right? So it's really three different ways of looking at it. And then, of course, if you believe in the whole spike, in the stools as well, possibly, is that causing some problems down the road in the colon? So, I think long COVID is not that straightforward, but to me, it's gut dysbiosis. We've published on that. We did an abstract that we presented last year at ACG, where we showed long COVID patients were lacking certain microbes and had an overgrowth of other microbes. So it is a leaky gut. It is a dysbiosis. Whether it's caused by the virus or caused by the treatment that you took for the virus, that's, who knows? You have to take a good history from the patient.
For sure. I think there's a long COVID and post-vaccine long COVID. I think
yes.
Probably two separate syndromes.
Well, it's interesting because I think the important thing to kind of take in the history of the patient, because I have people that come in and say, I have long COVID. And then you take the history and you say, well, when did you have COVID? Oh, I had it in January 2022. When did you get vaccinated? March of 2021. Oh, okay. So you got the vaccine that was supposed to protect you from COVID, but you got COVID.
Yeah.
And therefore, I don't know that that's a long COVID. I think that was the beginning of the demolishing of the spike protein from the vaccine that started that. So, and we'll discuss that later. We discovered that the spike protein kills the bifidobacteria, which is an important microbe.
Oh, right.
And so, when you start taking the history of these patients and you say, well, you had the vaccine, our data showed that you could, up to 9 months later, the spike protein keeps killing the bifidobacteria. So when a patient tells you, well, I got COVID a month after my vaccine, or I got COVID 9 months later, and I kept getting COVID, is that a vaccine side effect, or is that COVID side effect that caused the dysbiosis? I challenge to say that it started off with the vaccine causing the problem. So to me, that's how I differentiate them.
So there could be two pathologies going on here. Presumably, if there's active virus being excreted, what did you say, 9 months later or something?
Yes. So you can have COVID in the stools up to a year later. That's the longest we found.
Right.
I'm sure it's in others, but...
So the virus must be reproducing inside the cells of the gastrointestinal tract somehow.
Yeah, it does. There's been data in GI that shows that it is persisting. So, it's not just Sabine Hazan saying it. So there's definitely data out there that it is persisting, it is reproducing. Listen, we tend to think of our poop as just poop, but these are live microbes in there, right? We have live microbes that are living in our guts and are doing some function. They're either there to, break down sugars. They're either there to break down calcium. So these microbes are very well alive in your gut. Now, what happens when you introduce a virus to that live environment of microbes? For the most part, it'll start what's called a bacteriophage, killing those bacteria. Remember, a virus is 20 times bigger than a virus. So, you've got these big bacteria that are basically getting killed by an overaccumulation of viruses in the gut. It is causing problems, and it is causing what's called a bacteriophage. So, and in the case of bifidobacteria, it's causing a bifidophage.
Right, so in the same way that viruses can infect my cells,
yes,
viruses can infect bacterial cells. So a bacterium can get a viral infection.
A bacterium could basically get killed by a virus.
And that's the bacteriophage.
And yes, and remember, these microbes, interconnect. They're basically sequences of microbes, right? So, is a spike and that was the beginning of my questioning, right? At the beginning of the pandemic, we had an interesting case where we had 2 vaccinated patients. We discovered that they had, and actually Dr. Robert Clancy, who was here in LA, looked at that data and was really, fascinated by that because, and asked the same questions I did, which is, how is it possible that in 2021, it was in July 2021, we discovered the original Wuhan strain in 2 patients that were vaccinated. So remember, by the time the virus came to America, it had already passed from China to Europe to Italy to America, right? When it got to here, it was already, we were 4 or 5 spike mutations in the virus. When you see a virus with 3 parts only, that's the original Wuhan strain. How does that happen? I propose the hypothesis that perhaps the spike protein entered the blood, traveled to the colon, found an old coronavirus from, I had a coronavirus in my gut before the pandemic. You go to bat caves, you're going to be exposed to coronaviruses, right? Now,
but there's 7 human coronaviruses anyway.
Correct. So now, is there a coronavirus that is basically activated by that spike protein that was mimicking the Wuhan strain. I mean, if you think back of the whole way, and I think everybody by now realizes, we're in the world of designer bugs, right?
Yeah.
They have the capability to take tuberculosis and a virus and put it together and create these designer bugs, right? So this was a design bug. This was a designer bug. This was a creation of sequences, right? That you basically say, I'm going to take A, B, C, and I'm going to join it with D, E, F, and I'm going to create a novel microbe, because now I can patent that novel microbe. And now I can have a treatment that's a vaccine that's A, B, C, D, E, F that matches that novel microbe, and I can license that novel microbe. I mean, this is the world we're living in right now.
It's quite a good business model, really, isn't it? You invent a new bug, then you invent a vaccine to treat it.
Well, because you cannot patent bugs. You cannot patent known microbes. You cannot patent Clostridium difficile, for example.
But you can patent vaccines.
But you can patent a combination of Clostridium difficile with a Clostridium perfringens.
I see. This is worse than I thought. So I knew people were jiggly pokering around with viruses, but they're actually combining viral genetic material and bacterial genetic material.
Yes, yes.
But that's going to, I mean, that wouldn't happen in nature normally. It's going to produce something completely new.
And unfortunately, this is the world we're living in. And this is why we need to stop that, because we are creating superbugs, and that's not going to be helpful to fix this problem, right? Because, listen, the body rejects the unnatural, but up to what level? Certain people will survive. They have a resilience in their microbiome. They're resilient. Evolution, the strongest will survive. But how many will die from those experiments, right? And yes, you could do an experiment just in case a microbe does this with another, because that's the reasoning, right? In the mind of a scientist. Well, we better experiment to see what's going on just in case it happens in nature later on. Yeah. But here's the problem. You're creating the just in case it's going to happen in nature down the road, right? I mean, it's like, I hear scientists saying, oh, wouldn't it be cool if we could reproduce a dinosaur? No, it would not be cool to reproduce a dinosaur, okay? I mean, yeah, if you're, smoking something and you want to see a dinosaur, but the reality is, what is that dinosaur going to do once it starts growing up? It's going to kill humanity. So I think we tend to push these experiments, and there's a lot of scientists in their labs right now doing a lot of crazy stuff, and we need to really have better supervision of all that.
Absolutely.
So, yeah.
Just tell us what a dysbiosis is, Sabine, please.
So dysbiosis is an imbalance of the microbiome. It's, I don't like to think of bugs or microbes as good or bad, because I really think they're good up to a certain level, they're bad to a certain level. But you have to imagine that if you have an overgrowth of a microbe that is supposed to break down sugar, and then you have an undergrowth of a microbe, like you've lost the microbe that breaks down sugar. And now you have another microbe that's doing something else, but it's too high. That imbalance between the microbe that's overgrown and the microbe that is useful to break down sugar is really the dysbiosis. We're going to come out with a definition, and that's one of the projects I'm doing with Dr. Clancy, is basically and also a scientist that used to work at the National Institute of Standards, to basically look at all the data. We have thousands of stool samples of patients with diseases, because we're conducting 61 clinical trials on the microbiome. So my goal, I know. So, you think about, what does the microbiome look like in diabetes? What does the microbiome look like in alopecia areata? What does it look like in, pemphigoid disease? What does it look like in lupus? So all these diseases, we wrote 60. Remember, I'm the girl that was doing clinical trials for pharma. So I have about 300 protocols in my closet, in my storage, from different conditions. So we were able to kind of compile all these protocols and make them specific to the microbiome and certain diseases. And then from there, we basically are trying to map the microbiome and diseases. From there, what we've noticed is that there is something that we call dysbiosis, which is an imbalance of the microbiome. So that's what dysbiosis, leaky gut that you call is. You have to think of your stools as the first wall of protection. You know, when you ingest things that have viruses, fungi, parasites, your stools is really what's your defense, your first defense. Then you've got your colonic mucosa, then you've got your blood vessels, then you've got your nerve. So if your stool, your barrier is gone, those microbes are passing, and you have leaky gut, those microbes are passing from the colon to the blood vessels, to the nerve. They travel, you know, they secrete metabolites, which basically then go to the brain, causes problems. And that's what we're starting to see with Alzheimer's, Parkinson's, MS, ALS, these microbes that are basically out of whack, and, you know, metabolites going up and down and causing nerve damage.
So when I was young, the idea was that the gastrointestinal tract was basically this barrier that large molecules like proteins and things couldn't get through. But that's wrong, isn't it? This idea of a leaky gut.
You have to start at the microbes. You know, we trained, and you and I, probably the same generation, we trained with cytology, right? Remember cytology, histology, and we trained with mitochondria and Krebs cycle, and that's how we learned, right? It was all at the cellular level. But before the cellular level are microbes. So before you even get, so I like to, if this is my hand, if this is my colon, right? And I'm ingesting sugar in my gut, and now I have my sugar goes into my colon. There's microbes in that colon that are helping that sugar break down to penetrate, to let it go into the cell. Then what's in the cell, the mitochondria is basically using it. The Krebs cycle is using it. Function of the cell happens. But before all that are the microbes that are doing their job. Now, if you are lacking those microbes that are breaking down your sugar, that are breaking down your calcium, you're never going to put that sugar in the cell. Where's that sugar going? Everywhere else but the cell.
So the dysbiosis can actually affect the wall of the gut, so it becomes leaky. Does that mean things like large molecules and even parts of bacteria can get from the lumen of the gut into the blood and therefore make you feel sick?
Well, that's the concept, right? That's definitely the thought process, the hypothesis. That's why you start seeing people that have chronic UTI. The work of Dr. Sahil Khanna at Mayo Clinic, he did a lot of work looking at people with C. diff that he did fecal transplant, and they had chronic urinary tract infections. And when he restored the gut, he improved the chronic urinary tract infections. Did the chronic urinary tract infections occur because of that leakage, because of that loss, because of those lost microbes, right? We tend to think as microbes as bad, but we also forget that microbes are good. And so when you kill the bad, you're killing the good as well, right? Remember, during the pandemic, what did we do? We were so, we were hunting the virus, and we were all afraid of the virus that we ended up taking all these meds that were killing the virus, but also killing the microbiome. And so, yes, your microbiome will come back, with time and good nutrition, etc. But unfortunately, the damage is done. We think of microbes as bad and causing a problem, but we don't think of the lost microbes causing the problem. And that's what I'm trying to really get all the doctors to start thinking of. Hey guys, stop killing. You've killed enough. You think of all these. I give lectures at the ILADS, Lyme Disease Association, right? And I speak to all these doctors I'm the missing link, right? Because everybody, kills at those meetings to basically kill the bug, right? Kill the tick. And they forget you need to replenish the gut. You need to start focusing on healing the gut because you've just killed too much. And in time, we're going to have better diagnostic tools for Lyme disease where we could say, okay, you know what? We've killed the bug. Now we got to replenish the microbiome.
Mm-hmm.
You know, it's kind of like C. diff. We give vancomycin for 10 days. We replenish with a new microbiome, and then we let it be. We don't see these patients ever again. So
if I wanted to get a dysbiosis, I would take antibiotics.
that's one way. You could get a dysbiosis by drinking a lot of alcohol. You can get a dysbiosis by taking on a lot of products out there that are actually, you know, not as good for your microbiome. You know, there's so many things out there from, you know, in the nutraceutical world even that can kill the microbiome. Think about the preservatives that they put in all these products to preserve them. Is that killing your microbiome? Think about the capsules. What kind of capsules are you putting your vitamins in, right? So we were the lab that showed vitamin C improves the bifidobacteria, the good bacteria. If you have a capsule that is of a certain substance that kills the bifidobacteria, it doesn't matter that you're giving it vitamin C because you're now killing it with the capsule itself.
So the material the capsule is made from.
The material that you're making the capsules from is important. The contaminants is important, can kill the microbiome. Like I said, alcohol, high consumptions of alcohol, drugs, certain drugs kill the microbiome. You know, certain products kill the microbiome. So there's a lot out there, and we're just kind of tapping the surface of what kills and what heals. And I think as we get into this world of the microbiome, it's important. And that's really my work, is really to see what is Manuka honey doing in the microbiome? What is apple cider vinegar doing to the microbiome? What is cumin doing to the microbiome? I think we really need to start looking. You know, it's one thing to look at mice studies and animal studies, but they unfortunately do not reproduce in humans. Mm-hmm. We really need more human studies on all these natural products to really, because here's the problem. When I'm treating my patients, I'm trying to regrow microbes. If I give it something else that destroys the microbiome, then it's not going to help. Absolutely. You know, it's like putting water on a fire and gasoline at the same time. You're never going to turn off your fire.
Trouble is, there's not much money in honey, is there, really?
No, there's not much money. But we don't know what honey is doing. Actually, I'm excited because we're doing a study on honey.
Or apple cider vinegar. I mean, what I'm saying is these things can't be patented, so there's no impetus to put big pharma money into it.
Well, but I'm doing it, so I don't care. I want to know.
So do I. Thank you.
I spend more money on stool studies than anything because I have a curious mind and I want to know. And ultimately, like I said, I cannot reach success. And we're gonna talk about a certain, bunch of cases that I'm treating by manipulating the microbiome. I cannot reach success if I don't know these things, if I don't know what all these drugs are doing on the microbiome. There is a model that was done. I'm excited because I'm working with this new scientist now, and he told me about this colon model that Harvard developed, where you basically don't have to wait to enroll patients. It's kind of like an in vitro model, where you basically give the product and see what it's doing to the colon, in essence. It's a colonic model. So I'm excited to work with that because it's going to speed up the research to do the in vitro study first. Because think about what I do now. I'm taking my patients, I'm giving them the same exact protocol of food, and I'm changing, I'm giving them a lot of Manuka honey after a week and then retesting, right? So it takes a month to get those results, and then it takes another 6 months to a year to gather at least 20 patients. It's much faster to do it in vitro to begin with.
But presumably Californian honey is pretty good as well, is it?
Yeah, I mean, it should be, hopefully. We have a lot of problems here. We have fires, we have mudslides.
Yeah.
So I don't know. I do have 10 beehives, so I can tell you I have my own honey. Yes.
Fantastic.
I'm all about protecting the bees.
Oh, absolutely. Yeah. We won't start on that one. We agree on that. But
yes.
How much vitamin C do you take personally?
Well, during the pandemic, I was taking between 1,000 to 3,000 milligrams.
Right.
I don't take anything anymore. I've kind of balanced my microbiome. I'm a little bit privileged in that I have a lab that can test. So I try to,
Yeah.
I always see, okay, I'm engrafted. I've got some good bifidobacteria. I'm good. I don't need to worry about it. And it's also summer. So you remember you're outdoors, you're exposed to microbes, you're essentially it's much better, and we're getting vitamin D from the sun, which also, by the way, increases bifidobacteria. So
does it? Right.
yeah, so you don't really need as much.
Maybe in California you get vitamin D from the sun. We don't get too much.
Oh yeah, where you are, yes.
Yeah.
So where you are, probably increase the vitamin C, yes.
Yeah,
But
and of
between
course,
1,000
the older you get, the less your skin makes vitamin D, of course. So,
I think the older we get, the number one vitamin people need is vitamin D
absolutely.
and K2, of course.
Absolutely, yeah,
Yes.
absolutely. So you're giving these fecal transplants. Let me get this right. That is a probiotic, isn't it?
I guess you could think of it that way. Probiotic is really a couple of microbes, the bifidobacteria, the lactobacillus. Fecal
Right.
transplant is really a consortium. It's like 10,000, 15,000 microbes that we've identified that we put in. It's literally the stools of a healthy individual.
So when I'm eating fermented foods and foods with bacteria
Yes.
in it, which is good, the lacto and
Yes.
the
Yes.
bifidobacteria, I was always taught that the hydrochloric acid in your stomach kills it all.
Yeah, and some of it passes. Remember, the fermentation is really the products of the microbe that you're getting that's feeding the microbes down the road in the colon. So when you're eating something, when we think about, and I'm going to talk about ivermectin for a minute, even though it's a taboo word. What is ivermectin? It's a fermented product of a bacteria, right? So it's essentially the metabolites, right? So that bypasses the acid and goes into the colon. So it's really the microbes themselves that can get killed in the stomach. So, yes.
So what's going through? Is it the? Sorry, I haven't quite got what's going through the stomach. Is it the bacteria themselves
The
that form
metabolites
spores?
are going down. The metabolites are passing through the bacteria. Some of it passes through. Remember, you're
Right.
eating food, so have you neutralized your acid? There's a lot of component. When you're drinking milk, you've neutralized, your acidity. So when you're drinking kefir, it's an alkaline
Right.
product. So the microbes are going into the stomach and they're going into the colon, but you've kind of neutralized your stomach acid at that point with the product. When I'm eating a yogurt, the yogurt itself is not acidic. It neutralizes the acidity in the stomach.
So,
And also remember
a few percent of the bugs can get through.
Yeah, I mean, but is that the way to replenish, to recolonize? No. I mean, you can eat all the yogurts, because everybody asks me, well, what yogurt should I eat? What probiotics should I take? The reality is that doesn't engraft. It doesn't go, you're right. It does get killed in the stomach or gets killed in the small bowel, or it never really reaches the colon, or very few reach, and you really need to implant. When we do fecal transplant, that's a different ballgame. Remember I told you, we clean the colon, we look at the mucosa, we implant directly. That's a different process than just taking and swallowing probiotics. Now, some people will get lucky and the probiotics will land where they need to land and implant, and that's great. But the majority of the time, and the problem is, when do you stop taking those probiotics? Do you keep taking those probiotics? Very few probiotics engraft, meaning implant. You got to keep taking them to sustain. So it's a difficult process, the whole thing, and it's hard to heal that way.
Right. So, what's more effective, to give PR fecal transplant, or Dr. Broder's so-called crap caps that go all the way through?
Yeah. So fecal transplant, remember, is only for C. diff.
Okay.
Only.
Right.
So if you don't have C. diff, you can't get fecal transplant. Now, you could probably get it in another country, but here's the reality. We are not there yet. It's exciting. We're seeing 2 cases of Colleen Kelly with alopecia areata. We're seeing a few cases of Dr. Sahil Khanna, but we're not there to say we should do fecal transplant for chronic UTI. We should do fecal transplant for alopecia areata. Because here's the thing, the 2 cases that improved, for those 2 cases that improved, I have 2 other cases that went to other countries and basically lost their hair from the fecal transplant. So for everything that heals, that same thing can kill as well. So when I say, the work that I'm doing right now on those clinical trials is so important because it's really helping us fine-tune what are we seeing in the microbiome when we do fecal transplant, when we achieve a cure. We go back and see what changed in the microbiome. Let me tell you, when I stepped into this, I thought autism, and I'll just give an example of autism because autism is really what got me into this. I went to Dr. Feingold after I improved my 1 case of Alzheimer's with fecal transplant. And I said, Dr. Feingold, he wrote the book on anaerobic infections. And I said, what am I seeing when I'm improving Alzheimer's using the wife's stools? What am I seeing? He goes, you're seeing a microbe I've been culturing for years. Here's a paper I've almost published, didn't publish, put it in a safe. When you buy your machines to analyze the microbiome, you're going to find this microbe, and then basically that's going to validate what I've been saying. Okay, so I took the paper, put it in a safe. I said, okay, well, I didn't really think I was going to open a genetic sequencing lab, right? But here's what happened. He was very big on autism. Dr. Feingold is the first one to basically start talking about vancomycin for autism, fecal transplant for autism. Him and Dr. Barodi actually had a patent together for fecal transplant for autism. So when that happened, I started working with him. All of a sudden, I had an autistic kid that was 17 years old show up in my office. They heard I was doing fecal transplant. They read about fecal transplant for autism from Dr. Barodi. And basically, the kid was banging his head, nonverbal, in pain, breaking his teeth. And I felt sorry for the family and for the kid. And I said, I'm going to try to help this family. And I'm going to push a fecal transplant case with the FDA, because everything, if you want to do fecal transplant in America, you have, and it's not C. diff, recurrent C. diff, then you have to get the FDA involved. So if I want to do, I did a case of metastatic mesothelioma with fecal transplant, I had to get the FDA involved. I did a case of autism, I had to get the FDA involved. Now we're getting, Parkinson's and more autism, we have to get the FDA involved. So these are not easy cases. And remember, these regulatory guys that we hired to write these protocols and go through the FDA, they're $400 an hour. So this is not easy to push a protocol through the FDA. But nonetheless, when you do push it, when you do the case, you start. So this kid, when I started, I looked at the whole family's microbiome and noticed his sister could be a good donor, used his sister as the donor. And I saw it change. The kid started speaking. He stopped banging his head. He stopped being aggressive. I saw it change. Now, Dr. Feingold had said, autism is the sulfovibrio, is a bacteria called the sulfovibrio or Clostridium perfringens, right? So I'm looking for...
They're the bugs that cause it.
That's what he believed. He believed that autism was an overgrowth of those bugs, right? The sulfovibrio or Clostridium perfringens. Yep. So when I started looking at autistic children, that was the first thing I started looking for. I was like, okay, where's the sulfovibrio? I don't see it. Where's the Clostridium perfringens? I don't see it. In this kid, I saw a bacteria called Lactobacillus animalis, and he had 48% proteobacteria, which now I know is way too high. And when I saw that, and he had 0 bifidobacteria, the good bacteria that helps, break down sugar. When I saw that, I said, wait a minute, so autism is not the same microbe. So do you see how complex it is? So, but what did I learn? I learned that I improved the kid, and then I could go back to my stool sample and see what changed. Now I'm adding another couple bugs to my to my differential for autism. I have a feeling we've seen a lot of different microbes for autism. So it's not that straightforward. And those are of the the microbes we know. There are thousands, if not millions or billions of microbes. We don't even know what they're called. So my thing is, now that I've analyzed hundreds of stool samples of kids with autism, is to look at that whole pipeline and to say, now let's look into the gray matter. Let's look for bugs that have the same sequence that don't even have a name. That's going to be fascinating, because now we're going to start seeing other microbes that are possibly linked with autism that we could say, if I recognize this microbe. So think about the field, right? We're trying to fix autism, and now we're heading into the world of fecal transplant because Dr. Adams is doing the capsules for kids with autism, which is much easier when you were saying which is better, capsules or colonoscopy. For little kids, it's not easy to do a colonoscopy. And so if a capsule can get you 20, 30, 40, 50% success, well, let's do the capsules, But here's the thing, there's 50, so if you look at the studies of Dr. Adams, he basically showed that 50% improved. He did 18 patients, and 50% of them improved their neurological and speech and ATEC scores and CARS testing, right? Which is the ways that we look for autism, right? But there's 50% that didn't improve. What happened to those kids? What's going on with those kids?
Well, presumably there's a multiple factor etiology in autism.
It's either multifactorial. So, for example, brain could be the primary. Nerve between the brain and the gut could be the primary. Gut could be the primary. But I think also within the gut, there's a compatibility. And I'm going to say it out there right now. I think there's a donor compatibility. And, there's no data on this. Nobody's talking about it. But why? But think about it. Why wouldn't there be a donor compatibility, right? We're not compatible to everyone in the planet. I mean, there's people that look at me and they can't stand me just because they look at me, right?
Me too.
And there's people that, and there's people that look at me and they love me, right? Is that a compatibility? I mean, and also think about the microbiome like what the blood is, right? Back in the olden days, people would bleed. In the days of George Washington, they would treat a cold by removing the blood of the patient, right? Remember, that's how
Absolutely.
they tried to fix George Washington, and then ended up killing him because they probably
killed him. Yep.
Took too much blood out and dropped his hemoglobin.
Killed by doctors.
Exactly, but we didn't know, right? Back then, we didn't even have what a hemoglobin is. We didn't have a CBC. Then came the idea of, look at that. Red blood cells, white blood cells, platelets. Let's quantify them. Let's say what a hemoglobin is. And then later on we said hemoglobin of 7 and below, people need to have a blood transfusion. The hemoglobin of 8 and above, you're fine. And here's the other thing that came on. Blood was not clean. Blood had hepatitis C. Blood had HIV. So we started saying, oh my God, there's infections in blood. We need to look. And by the way, I'm going to say, hepatitis C and HIV is one of the viruses that we look for, but there's probably other viruses that we're not even picking up. I have people that come in with neurological problems, and I test them for JCV, Creutzfeldt-Jacobs virus. And let me tell you, they have that virus. Did they get it from the blood transfusion? Does that virus cause a problem that we don't even know about? Does CMV cause a problem? Do we give CMV? Do we give herpes to patients when we do a blood transfusion? So there's so many microbes in the blood itself. The blood's not pure. So now multiply that by 1 million and think about your stools. You're literally giving microbes, and you don't know whether that microbe is good or bad, and whether you're compatible, right? So
the fact that someone's a sister or a brother or a spouse doesn't necessarily.
It's probably better.
It's a good guess. Yeah,
it's a good guess, but I've always done spouses. And I always joke, I say, be nice to your spouse. You never know when you're going to need their poop, literally.
Good point. Yeah.
I'm going to be polite on your show, but I usually say sh.t. So always be nice, but think about a husband and wife. They're pretty compatible, right? You've been married together. You're pretty much, having the same, you've shared microbes. You're compatible. I think that's probably why spouses are probably the best donors. And then you've got your children, right? Which child would you pick that kind of like is a young you, right? I know like in my family who I would pick as my compatible donor. So in case my husband doesn't work out, so.
Yeah. It's just the ecological interaction is just so complicated.
I think if we keep killing, and this is the problem that I have right now, we keep killing the microbes, we keep killing the bacteria, we keep killing the diversity, eventually the planet's going to be toxic, right? Mm-hmm. Because you're no longer able to survive. You're so sterile that when you go outside, the least amount of virus becomes toxic.
Yeah.
Think about 100 or 200 years ago, the diversity of humanity was probably in microbes was probably a lot more than we are now, right? If you think of so I tend to think of myself as just a reservoir for microbes, right? My body is just a reservoir. These microbes are doing what they're supposed to do. These microbes are giving me my personality. They're allowing me to function. They're allowing me to do whatever I need to do, right? But think about in the prehistorical times, the era of the dinosaurs. These animals were enormous. They had enormous body cavities. Did they have, trillions and trillions of microbes in there? And did those microbes slowly, disappear? And therefore, the body didn't need to be that big anymore, right? I mean, the evolution, when you think of evolution, are we shrinking up in sizes of reservoirs because the microbiome of humans, of the planet, of living things is disappearing? At least that's what I'm seeing. I mean, I wake up every morning doing this research, speaking, staying up all night on this, talking to you, mainly because I really want people to realize we're killing too much. We, out of 1,000 samples in the last probably year that we've analyzed, less than 5% had bifidobacteria, and 1 out of 1,000 stool samples had lactobacillus in them.
Both of which we believe are very good.
Very important microbes.
Healthy microbes, yeah.
Yeah, so what happens when bifidobacteria disappears and lactobacillus disappears? You can't absorb sugar, you can't absorb calcium. What happens to your Krebs cycle? What happens to humanity? Is this why we're having so much chronic disease? And I'm going to show you, in the slides if you want to go through them.
Oh, yeah.
We've discovered loss of bifidobacteria, this important microbe in so many diseases. So
I'm so interested in this gut-brain axis idea. Do you mind just sort of unpacking that a little bit for us?
So when you have a dysbiosis, an imbalance, microbes are either crossing, viruses can cross and go up and down, although that's debatable because nobody's really doing those kinds of studies to see.
From the gut up to the brain?
Yeah, but nobody's doing a brain biopsy to showcase the microbes. We all believe, like Dr. Jordan, myself, doctors that are on the front line, all believe that there's probably a microbiome of the brain, right, that gets altered. However,
mind blowing, isn't it? I mean, we've been taught that the brain's sterile.
Yes, well, I mean, yes, but is it sterile up to a point when you're essentially, when you have a dysbiosis, does it become sterile? Yeah. Is that why people have encephalitis? Is that why people have CMV encephalitis, herpes encephalitis? So it's not really sterile if you can have a virus in the brain causing an inflammation picture, right? And is it because of the fact that you've got, think about, microbes in the mouth now. They're starting to say microbes in the mouth are causing Alzheimer's, right? Because of that direct contact. So is the brain sterile, or maybe is the brain is sterile when you're healthy, but
Yeah.
then, and it probably has a healthy microbiome, but then when you introduce, you know, fungi, when you introduce viruses, it doesn't become healthy anymore.
Yeah, clearly you
So-
can get infections in the brain. I mean,
Yes,
you know,
exactly.
the encephalitis is a well-recognized
But I think that
pathology.
the infection, we have to start thinking about infection as an imbalance of the microbiome again.
Right, interesting. Yep.
So-
And does this affect mental health as well?
Yeah, so we actually just published, I'm going to show it later, a signature microbiome for anxiety
Yeah.
and a signature microbiome for bipolar disorder. So
Brilliant.
what's great about that is that now for the first time we can say, well, we've seen a marker for bipolar disorder, we've seen a marker for anxiety. Let's now fix the anxiety with whatever products out there. And let's see if that marker changes, if that dysbiosis decreases, right? If that gap, decreases. And that's basically, in a nutshell, how we are going to start looking at bipolar disorder. I will tell you that there are missing microbes in people that have bipolar disorders. So, did they lose it because they took antibiotics when they were kids? Did they lose it because they were traumatized? Here's the problem with mental health as well, is it's a two-way highway. The brain controls the gut controls the brain.
Yeah.
So if you've had trauma in your life and
Yeah.
that brain is affecting your gut, right? And vice versa, if you took antibiotics, if you, had killed your microbiome from the trauma, your microbiome affects the gut, the brain. So and those are the tough cases to fix, by the way, because you have to work with psychiatry, psychology, and GI. You can't forget the gut.
So prebiotics presumably are really important.
Yes.
The way that we feed.
Yes,
The
the
feeding
feeding of the microbes. Yeah, so they're
Yeah.
basically like the bed of the microbes.
So what are basically? I mean, is it possible to give sort
Fiber.
of a brief condensation of what good prebiotics are and what bad ones are?
Well, I tend to think of like fiber, right? Your
Yeah.
your fiber, your psyllium, your all those are really good.
All the fruit and
prebiotic-
veggie stuff.
fruits, vegetables. I mean, I love fennel, I love beets, I love all that as my prebiotics. So those are really your fiber. So you can never have too little fiber. I think the majority of people end up having problems because they underdo. Think about, we need between 25 to 30 grams of fiber a day.
Yeah.
That's a lot, right? That's like 4 prunes, 2 pears,
Yeah.
4 metamucils or citrusel, and
Yeah.
then, so it's a lot.
I remember in Tom Broder's clinic, he used to prescribe prunes. He'd say, right, take 6 prunes a day. It's just-
My father, I basically force. I have these bags of prunes, and I'm like, Dad, 4 prunes a day, 4 metamucil at night, 2 pears during the morning. So, and as you get older, the colon is sluggish, so you really need to kind of increase your fiber.
So it's all the basic things, really. It's the fruit, it's the veg, it's the beans, the peas,
Yeah.
the beans, the lentils.
So beans, lentils, I tend to stay away from in a
Oh,
person
okay.
that has a dysbiosis because they tend to be really gassy. So
Right.
beans, corn, chickpeas, I stay away from all that. They're very, very gassy, and most people that are dysbiosis come in gassy and bloated to begin with. So you really want to try to increase the fiber. There's a couple of good, without mentioning brands, because I don't like to be a salesperson. There's a couple good brands out there, and there's actually a lot of studies on prebiotic. Anybody can Google prebiotics, increasing bifidobacteria, and there's a lot of studies out there on products that have been tested to show increase in the bifidobacteria with prebiotics.
But whatever vegetables are cheap that week in the markets,
I mean-
that's good.
Fennel. I love fennel. Fennel, beets, red beets, sweet potatoes.
Beetroot. We call them beetroots. Yeah.
Yeah, those are great. Yeah, orange beets are my favorite. So, because the red beets, you can think that they change your color of urine and stools, and it makes you freak out a bit.
Well, you kind of get used
Because
to
you
it, I
think
guess.
like oh my God, I'm bleeding. Call the GI doctor.
Yeah.
Yes.
So, is there anything that's particularly bad for the microbiome? Again, in general terms,
Well,
artificial
stress. I
sweeteners.
think stress, artificial sweeteners. I like
Right.
fruits
Yeah.
and vegetables that I can peel. Let's be
Yeah.
clean.
Yeah.
Let's be
Yeah.
clear. They either have to be under the soil with all the microbes of the soil or on a tree. I don't like anything that grows above the. So I'm not a fan of blueberries, raspberries, strawberries. They're kind of like on the floor. And in California, they spray so much.
Yeah.
It boggles my mind
Yeah.
that
Yeah.
you've got this bottle of pesticide with a skeleton that say do not drink poison, and then they're spraying it all over the soil, and who's washing their strawberries that clean, so yeah.
Well, it's actually in the cellular structure of the fruit.
my
You can't just
God.
wash it off. We want the food, the whole food, and nothing but the food.
Exactly. And even the taste of the food is no longer the same. You've gone to you've grown tomatoes in your backyard and eaten tomatoes in the grocery store. Completely different taste.
Totally.
There's a sweetness to the tomato you grow at home. So it's kind of crazy. But
Yep.
I'll tell you a funny story. I was in Idaho fly fishing on a river, and it was in the middle of nowhere. Here I'm thinking I'm going to get the best trout, no pesticides anywhere around.
Yeah.
This trout is like in the middle of nowhere. And all of a sudden I see a guy with a hazmat suit spraying around the river. I'm like, well, so much for fresh fish. I got out of that lake. So it's so sad. They're spraying everywhere.
I hate chemicals. It's just
Ugh.
they're just so many, and it's so unpredictable what they're going to do to you. Right, that is amazing. Shall we look at one of? Have you got time to look at some pictures?
Yes.
So, what are we looking at here, Sabine?
So this is basically each column is an individual, and
Yep.
each color is a group of microbes, and this is basically showcasing 26 healthy individuals.
Oh, so each
And-
column is a person.
Is a person, and you could see how it's a signature microbiome. So if, let's say, patient 3,
Yep.
comes in my clinic and blocks the toilet, I'm going to know who it is based on his signature microbiome. So now this is what's fascinating is when you look at that, you say, wow, all these people are healthy, but they each have a different signature microbiome, right?
So each color is a
group
bug
of
that you
microbes.
can recognize. Yeah.
It's a group of
A
microbes.
group. Yeah.
So you have to say, well, how come patient number 3 has a lot of gray, which is like Bacteroides, and patient
Yeah.
number 2 has a lot of purple, which is probably some bifidobacteria?
Yeah.
But yet they're both healthy, right? That right away tells you we're all different. And because we're all different, we don't fit in a one pill, one shot solution for everybody. This is the beauty of diversity, and this is the beauty of humanity. How everybody is different in that cohort and healthy, and yet together they make a beautiful, diverse humanity. Now you remove a couple of, colors, and, you're basically globalizing humanity. So that's why it's so important to maintain that diversity of humanity, and not to think, well, we should all be like patient number 3.
Yeah. So
Right?
I'm eating a particular diet in the north of England, and someone in Amazon is going to be eating a different diet, and my mates in Africa are going to be eating a different one.
Correct.
So
And that's
there's going to be
again,
a lot of variability between the microbiomes.
and by the way, that microbiome is what allows the people in the Amazon jungle to survive the Amazon jungle. Think about it. You're surviving. You're in the Amazon jungle. These women live in the dirt with the plants. They eat from the plants and everything. They're not exposed to civilization much. They're deep into the forest. That microbiome is basically resilient to the environment. It's mixed into the environment.
So
you
it's
change the microbiome. Like, for example, you put me in the Amazon jungle, sterile girl, from California, have been washing my hands with soap my whole life, I go into the Amazon jungle, I can't survive. There's going to be microbes there that are probably going to cause me to have diarrhea, or that are going to cause me to have a skin problem, or I'm going to be, so these people that live in the environment, you cannot change their microbiome, because then they cannot tolerate their own environment. Think about
if you did change their diet, like if you started feeding them hamburgers and bottles of Coke and bottles of potato chips,
you're killing their microbiome, and they're not going to be able to survive their environment.
So literally changing giving them an American diet could literally kill them.
Well, look at what happened to Kiribati, right? The islands of the Pacific. We started sending them potato chips and Coke and American diet. Now the population is morbidly obese. They stop planting, they stop fishing. They're morbidly obese, they're having diabetes, and they're dying very young. So you are essentially killing that population because you've changed their diet. That population used to grow plants, used to, go fishing. They were more, energized. Now they're eating chips and potatoes, and they're on their phones. The worst thing that I heard recently is that they're putting, cellular data in the Amazon jungle. So these kids are on their cell phone. They're going to want to be like Americans, having chips and everything. So we're killing, in a way, the microbiome and the diversity of humanity by globalizing. And that's a big mistake, because, you really need that diversity. Like what I'm showing, these 26 patients, that's what makes the diversity of humanity. If we were all the same, think about, the same people, like 20 people like you, John. I mean,
Rather not.
No, I know. It's like me. I wouldn't want 20 of me. I mean, it'd be the world would go chaos. So, we really need that diversity, that difference. Those differences is what makes the planet and the world.
Yeah, the Australian Aboriginal population comes to mind as well. I mean, the amount of diabetes, the amount of alcoholism.
That's right. That's right. Sad.
Yeah,
Sad.
It really is.
So when you're all different, how can you compare? And so if you go to the next slide, what you can compare is families. And so the second slide is really, my family portrait.
Right.
And what you see is the two in the middle are very similar in their coloring of microbes. And the two at the ends are very similar, right? And so, it's kind of like, DNA. You share DNA, you get some DNA from your dad, you get some DNA from your mom. But even in the microbiome, you get some microbes from your dad, you get some microbes from your mom, and then you become this personality. Now, what's interesting is the two in the middle are identical in the personality and identical in their microbiome, and the two at the end are identical in their personality and identical in their microbiome. Now, that's a neurotypical family. You could see they're kind of scattered. There's nothing really popping up. If you go to the next slide, which is a family, this is a family where the first column is a mom, the second column is a kid with autism, the second column is a neurotypical kid.
Yeah.
Third column is a neurotypical kid, and the fourth column is a kid with autism.
All in one family.
Yeah, all in one family. So you could see this is triplets, 3 kids. One was autistic, the other 2 were normal, neurotypical.
Triplets who were clones.
Triplets. Non-identical triplets.
Non-identical triplets.
2
right.
girls, 1 boy, and the 2 in the middle were neurotypical.
Yeah.
And the third one, autistic, right?
And his mum on the left, is it?
The mom is the first one, the first column, yes. And what you could see, the FT0111, if you see, what you could see with that first column is there's an overgrowth of a certain microbe that you can almost say was passed on to the third child twice as much.
Right.
Probably in utero, or probably upon delivery, or maybe the kid didn't grab as many good microbes as his other 2. If you think about, I don't know if you raise chickens, but I raise chickens, and I have little chicks sometimes. And when you have chicks and there's always one weak one that basically dies because he's competing and he can't compete with his other the other chicks, and he can't get the food, right? So you wonder if in utero, this triplet didn't get as much of the nutrients and therefore ended up getting, autism, or got some antibiotics, etc., after delivery. But this is an interesting case because, they all had the same vaccinations, they all had the same antibiotics, and essentially 2 in the middle didn't have autism, and the third one did. This is where the idea of familial fecal transplant came from, which was basically taking from one donor and putting it into the other to fix the other.
I was going to say that dysbiosis is just begging for modification there, isn't it?
Yes. I mean, that's like the best way to kind of showcase it, to kind of see how we're doing it. Now, what's so fascinating is when we look at families like this, we really are able in our lab to say, okay, well, this is what I'm seeing. Let me correct this microbe, because really in the microbiome space, it's about finding the microbe that's overgrown, getting rid of that microbe, and then repopulating. You can't really repopulate if you don't get rid of that microbe to begin with. So it is a complex, it's not just, that's why I'm saying there's a donor, specific component that's going to have to be looked at in the future.
So it's amazing. I mean, when I visited Tom Brody, he showed me some people. Well, one guy I went in, and he was sitting in the cubicle, and Tom says, "Well, this guy's basically fine now." But he'd actually been in a hospital in London where they were going to do a total colectomy for his. I can't remember if it was colitis or Crohn's. I think it was colitis. They were actually going to take this poor guy's colon out, and Tom fixed it.
Yes, I know. Him and I, we have these discussions. We had and have these discussions where we basically are laughing because we're like, how barbaric. They're still taking out colons of people. I feel like I'm, the doctor in Star Trek where you remember that episode where he's going by a patient and he's saying the kidney dialysis, how barbaric. Take a pill and you'll fix yourself, right? And that's really, I used to show that video because I used to say, and that is the pill poop.
Yeah,
but,
absolutely.
Maybe in the future, you never know. But yeah, how barbaric that we would take colons off kids. I recently had a kid that came into my office. She was having rectal bleed. She was heading into the path of Remicade, biologics, etc., Crohn's disease. And basically, I treated her. We found the microbe that was the problem. We treated the microbe, replenished her gut, and the kid is done. She's in remission, and she's so much better. And this kid was heading into the path of, at 4 years old, of biologics after biologics, pneumonia, ended up in the hospital. So, unfortunately, what we know of medicine right now is still backwards, right? Think about what I do. I do clinical trials for pharma. That's already 20 steps ahead of every other doctor. And then you add on top of that, I'm into the microbiome space where I'm trying to figure out consortium of microbes to fix disease, trying to understand, diseases, period. So I'm in the research world, which is already 20 steps ahead of every other doctor, because back in academia or back in regular practice of medicine, the guidelines are just not there. They're not even looking at this stuff. This is so ahead of its time that,
but people like you and Tom are putting it into practice. I mean, this guy I saw who was cured, basically, Tom had just washed out his colon, recolonized it.
Yes.
And his colitis was gone, and he was, rather than having a total colectomy, he was normal for the rest of his life. I mean, this is available now.
It's available to a certain level. It's definitely not available in America because, like I said, for Crohn's disease, you have to get an IND.
Which is why some rock stars flew to Australia to get treated by Tom Brody.
And by the way, my patients as well would fly to Australia to get treated by Tom Brody because exactly, we're kind of, we're handcuffed here, unless we have C. diff. But I think it's going to have to be moving faster, right? They're going to have to speed it up because the public is going to ask for it. The public is tired. They're tired of the old science. They're tired, antibiotics were great.
And still are for the right thing.
Yeah. And listen, I mean, I trained in the world of antibiotics where we were giving antibiotics for everything. Psoriasis, antibiotics, rheumatoid arthritis, antibiotics, and then came the biologics. And then it became biologics for everything. And now we're in the pill poop level. And it's going to be pill poop for everything, So science is only good as science is during the moment in time where the research is not advanced. What me and Dr. Barotti do is we're the innovators. We're the ones that are basically on the front line challenging the status quo and saying, why not look for this? Why isn't Crohn's mycobacterial paratuberculosis? And why shouldn't I look for it? And should I be giving antibiotics and then implant? So there's so many different causes, but I think you have to play detective and you have to innovate and be outside the box. And there's not many doctors that are willing to do that or be that.
And the few fortunate patients that come under your care can be cured of conditions which otherwise they would die from or be subject to brutal treatments.
Yeah, but my patients understand. What I do is research. I'm extremely regulated. Everybody's watching me. Yeah. So, but that's why I stepped into the front line with COVID. COVID, by the way, was a great opportunity to see the microbiome, to understand the microbiome. So.
Yeah, because you were looking for the virus in the stools.
I was looking for the virus in the stools. We found the virus in the stools, and then when we looked for the virus, we asked, what is the microbiome doing when COVID is in the stools? And that's when we discovered bifidobacteria. That's when we discovered this important microbe that basically, helps break down sugar, that helps with immunity. And that is your $1 trillion industry of probiotics. So really, COVID was the beginning. But look, I had to do that with the regulatory boards. I had to have FDA in my office. When I took on the protocols with Dr. Barotti, we did the ivermectin, doxycycline, zinc protocol with vitamin C, D. And then we did the hydroxychloroquine, Z-Pak, vitamin C, D, and zinc. These protocols were written with the FDA, approving those protocols. So we were really essentially the only ones treating with the FDA watching to write the data. It's really a shame what happened to both those protocols.
Can we just mention those protocols? Yes. What was the first one?
Hydroxychloroquine, Z-Pak, vitamin C, D, and zinc.
What's Z-Pak?
Oh, azithromycin.
Oh, right. So I've forgotten already. So it's hydroxychloroquine.
Yes.
Erythromycin.
Azithromycin.
Yeah.
And then vitamin C, D, and zinc.
Vitamin C,
D,
and zinc. Yeah. And that's for acute COVID.
It was for acute COVID. What I came to realize, it was for the severe acute COVID, like the person that you already assumed had very poor microbiome to begin with. You wanted to kill the virus as quickly as possible. That was the purpose of those drugs. When we started giving ivermectin, doxycycline, zinc.
This is the second one.
This is the second protocol we wrote, and we got approved to run it in July. We actually finished that protocol. We're going to publish on it. And it was very successful because it was a placebo-controlled trial where the placebo was placebo. And the people that so we had an enormous amount of people that succeeded with the real drugs. And then the placebo, we barely had 13%.
So the trials that proved ivermectin didn't work, the people that conducted those trials, I'll have some questions to look at, won't they?
They're going to have to, because our trials worked. And by the way, not only did my trials work, but no one died during the pandemic.
Your ivermectin, doxycycline, zinc trials
both, and the hydroxychloroquine, Z-Pak.
Also worked.
In the sense that if they had placebo, right, and they crashed, we treated them with ivermectin, doxycycline, open label, right, when they crashed, and those people survived. No one died on my shift. I was conducting 3 clinical trials, placebo-controlled trial, during COVID, and no one died. So don't tell me treatment didn't work when no one died on my shift. And by the way, we're not talking about, and that's why I did it with the FDA, by the way. I did it with the FDA to say, you're going to watch me, and you're going to watch that everyone's going to survive. And not only that, I'm going to put myself on the front page of the newspaper interviewing those people whose oxygen were in the 70s and 80s and who survived at home without oxygen, because that was the power of ivermectin. The power of ivermectin was that it increased the bifidobacteria within 24 hours and therefore decreased the cytokines and essentially improved the oxygen saturation of these patients. I lived it. I saw it. I was in communications with Dr. Barotti. We were like sending each other, I was sending him texting of oxygen saturation, 63%. And then 5 days later, the guy's at 92%.
This is exactly what Jackie Stone found in Zimbabwe as well.
And by the way, Jackie and I were communicating. And it's unbelievable to me that nobody even tested that. But I'll tell you who tested that. The patients.
Yeah.
The patients who took ivermectin, the patients who had COVID, had their oxygen drop, and then took ivermectin, they saw their oxygen. That's why it became a healthcare revolution. It wasn't Dr. Hazan or Dr. McCullough or Dr. Pierre Corey. Sure, we leaked it. What we did, we kind of were talking about our experience to try to see if others, but let me tell you, that word of mouth PR of ivermectin saved so many lives and created a distrust in the industry where people woke up because they said, wait a minute, my oxygen was low and I did take ivermectin and it did work. Why are they telling me ivermectin doesn't work?
It's really hard not to get angry that the WHO trials and I think it was the Oxford trials demonstrated that ivermectin didn't work, but it patently does. There's some fundamental problem with the way those trials were conducted.
Yeah, I will tell you the single fundamental problem. They gave hydroxychloroquine. When they did the study on hydroxychloroquine, they gave it with food. You're supposed to give hydroxychloroquine on an empty stomach.
Right.
And with the ivermectin, they gave it on an empty stomach. You're supposed to give it with a fatty meal. It's even in the Merck when they brought out ivermectin initially for scabies and lice. And by the way, my sister was the one that brought ivermectin to the market doing for lice.
Really?
Way back, yeah. She used to advertise. I think she got she got hundreds of patients on that clinical trial.
But the idea that the leading clinicians in the world, allegedly, would give these drugs without knowing. I mean, they should have known that you give it with food. You give hydroxychloroquine on an empty stomach and you give ivermectin with food. Well, why the heck didn't they know that? They must have known that.
I mean, I don't know. Physicians follow guidelines. And physicians follow the rules of the hospital, and they're afraid of getting sued. And this is what we did with our medical community. We handcuffed the doctors. We made them scared of a lawsuit. We made them scared of losing their license. And therefore, they stop. They don't even want to know. Okay? And
but the ones doing the research can write the protocols. They're the ones that
Well, I mean, listen, me and Dr. Barotti, we had scientists in our offices that were doing the research for us, because I'm not going to write a protocol without knowing all the data. Yeah. There are doctors out there. Peter McCullough was one of those doctors that was reading the data. Lynn Finn was another doctor that was reading the data. So many doctors in Australia, they were reading the data. When you stop reading the data, and by the way, you could read the data that's presented on CNN and Fox, and you could read all the data. I encourage doctors to read all the data. The good stuff that's presented on CNN, the stuff that's presented on CNN and Fox, and the stuff that's not presented. Go do a PubMed search. And especially now today with AI, although I have to say AI is also going to be flawed because they're retracting so many papers to make AI be a certain narrative. So you'll never see AI say treatment worked. What you'll hear AI say is, Dr. Hazan was controversial during the pandemic because she said treatment worked, right? Because they removed all the papers that said treatment worked. And here's the other thing. You know, science is about prove me wrong.
Yeah.
Prove me wrong.
Yeah.
You know, make a fool of myself that I was wrong, right? Show me another paper that shows Dr. Hazan was wrong.
Yeah.
But retracting it when it's not a fraud paper is just wrong, okay? Because, listen, there were a lot of things in medicine that we've proven wrong. But that's part of history.
Yeah.
You have to go back and say, oh my God, look, doctors were so barbaric back then. They used to operate without washing their hands or put on gloves, right? That's why we do research to advance. But we need the history of what went wrong so we can never go back to that. Science is about, again, you know, prove and disprove, prove and disprove.
And we've already done that. We've learned from the mistreatment of George Washington.
Exactly.
As we talked about
Yes, exactly.
earlier in this talk.
But there's a lesson there. That's the history of medicine, right? Medicine is not perfect. We don't know it all. We have to stay humble to realize, hey, we made mistakes. Listen, antibiotics, like we said, are good for the most part, but in some people it causes C. diff.
Yep.
What are you going to do?
Yep.
You know?
Yep.
It's a risk-benefit ratio. We have to start practicing that way.
Yeah. Just out of interest, will most people with COVID respiratory infection excrete the virus in their stool as well, or is it a minority of patients?
No, we actually, we tested a lot of patients where we tested the respiratory and the gut. All the people that had respiratory
So
had
I guess the
COVID
swallowing
in
it,
their
aren't they,
stools.
is going through.
The rest, yeah. Well, you're swallowing, it's going through your blood vessels travels to the gut. But here's the interesting thing. It is cleared in the upper respiratory first, and it is cleared in the gut second. Here's another interesting thing. Majority of people, and I'm going to say it because I cannot publish it, the majority of people, it starts in the gut first. You're going to the bathroom, you're having diarrhea, the stools, you know, evaporate some virus, and then you breathe in the smell of the diarrhea. And then you get the upper respiratory. So when you're on an airplane, you're actually catching the virus, not from the guy next to you that's coughing. Yes, you could probably catch it from the guy next to you that's coughing, but more likely you're catching it from the person from the toilet on the airplane. You know, that's the person's going and having a bowel movement, and, you know, microbes are flying. You know, think of these little bathrooms in the airplane. That's really where you're
disgusting
going.
thought, but yeah, it makes sense, unfortunately.
Yeah, so I've analyzed the stools of thousands of patients with COVID, and I can tell you there's a smell to COVID. Like there's a smell to C. diff. And in fact, you know, they have like these little dogs that sniff. I can tell you, I can go to the bathroom in the airport and I will tell you someone
Really?
with COVID was just there.
I can certainly do that with C.
Oh,
diff.
yeah. C. diff. C. diff. A patient would come into my office, and I don't even need to take. I don't even need to take the history.
Yeah.
I'm like, you've got C. diff. I can already smell it. Even Parkinson's, by the way, has a certain smell. Parkinson's has a smell.
Yes, yeah.
So,
there was a woman
you know,
recently who
all
could
these
smell
microbes.
Parkinson's, wasn't there?
Yeah, no, Parkinson's has a smell. I think, you know, these microbes are, you know, secreting toxins and they smell.
And the reason that diseases have a characteristic smell does really beg a microbiological explanation, doesn't it? Because I mean, I can tell by sniffing, you know, you can, I can tell some bacterial infections by smelling the wound.
Yes.
You know, you can recognize Pseudomonas from the typical smell when you're dressing a wound.
100%. I have a very sensitive nose, and I'm starting to notice all these microbes. So I'm very sensitive to all that.
That could be put into a tricorder device that detects the different chemicals.
I think in the future, I think in the future it's probably going to be smell, you know, that picks up, you know, that's
because I can smell things because I've worked with wounds for decades, you know, but
yes, you have a sensitive, and you probably have become immunized in a way with all these wounds and all these infections that you're, accumulating. Think about what immunity is. Immunity is exposure little by little to microbes, right? Like I'm immune to C. diff. I'm probably immune to thousands of strains of COVID because I've been exposed to so many patients with COVID and stools with COVID that I'm not really, concerned.
Yeah.
I've yet to be sick. So, I think ultimately, understanding the microbiome and improving your microbiome is really where we need to start focusing. I think, the idea of vaccines and everything, it's like antibiotics. They had their time, they have their time, they have their roles. Yeah. However, we need to keep advancing. We need to keep advancing science, and we need to say, okay, well, when I think of cancer now, I don't think of cancer as, chemo and radiation and surgery. I think optimize the gut, focus on the gut, improve the gut, because these people are going to die from viruses. That's what's going to kill them. Infections. Think about your cancer patient who's on chemo. What's going to kill him is not the tumor. What's going to kill him is the killing of the microbiome, which made him susceptible to multiple infections, and now he's having fevers and chills, and then he dies of an infection.
Absolutely. It must be getting near your bedtime, is it? Do you want to do some?
Yes,
shall we leave it there for today? I know this.
Yeah, we'll leave it there,
so
and then we'll come back
just
for another day.
We'll just tantalize people with the next slide that we could maybe do next time. But
yes, if COVID is in the stools, what is it doing to the microbiome? And what does the microbiome look like?
We'll do that next time. It's definitely your bedtime. Thank you so much for today. That's getting on for two. That's an hour and three quarters. So we'll put that together first. And, fascinating area. Just thank you so much. And really, this is a publication of sorts because,
Yeah, I mean, it helps people understand and ultimately listen. I think the world is changing. We've got to revamp how we publish things. Maybe the publication becomes podcast, and thank you for the stage, and thank you for
it's just
giving me the opportunity to talk about it.
The world-leading experts choose to talk to me is just utterly bemusing to me, but it's pretty wonderful. But of course, students do now put YouTube videos. When I was teaching in the university, we worked out ways to reference YouTube videos, and that is, it's a reference. It can now be done. So it's now out there. Wonderful. Thank you so much, and thank you for staying up late.
Thank you so much.
Hopefully we'll talk soon. Thanks, Sabine.
Thank you. Thank you.
Bye.
Absolutely. Thank you.